So let's turn our attention now to the plasma membrane of cells. So recall that all cells have an external barrier that separates the intracellular environment from the extracellular environment, and that barrier is called the plasma membrane. So in our discussion of the plasma membrane, we'll first look at the structure and then we'll discuss its function. So the plasma membrane, as you can see from this diagram, is a complicated structure. It's composed mostly of phospholipids and other lipids like cholesterol, but it also contains proteins embedded within it or proteins that rest on its surface, and it also contains these chains of carbohydrates that extend from its external surface as well. So let's focus first on kind of the basic functions of plasma membrane and its basic characteristics. So the membrane functions as a physical barrier. So as I mentioned earlier, it separates the interior of the cell and the cytoplasm or the cytosol of the cell from the exterior or external environment. The plasma membrane is often described as a selectively permeable barrier, or it's also called, it's also described as being having selective permeability. So this term means, or this phrase means that some substances are able to cross the plasma membrane quite easily, while other substances are actually quite restricted from crossing the plasma membrane. It also functions in communication, as well as the proteins, or some of the proteins that are embedded with the plasma membrane interact with chemical messengers, and then they relay those chemical messages to the cell interior. And so you have these chemical messengers that will interact with membrane proteins. That chemical messenger will cause a change in the membrane protein, and that membrane protein will then have an effect on some cellular processes that are occurring inside of the cell. So even though that chemical messenger couldn't cross the plasma membrane, it still had an effect on, or it still influenced what was going on inside of the cell. The plasma membrane also permits cell recognition, as the carbohydrates that cover its surface allow the cell to be recognized by other cells. The basic structure of the plasma membrane is a bilayer that is two layers formed from a lipid called phospholipids. And so we have this kind of two-layer sandwich of phospholipids, and that composes the basic structure of our plasma membrane, or that constitutes the basic structure of our plasma membrane here. There are a number of proteins, as we saw in the diagram I showed you a few slides ago, that are associated with the plasma membrane. Integral proteins are actually embedded within the membrane itself, and these generally function as channels across the membrane so that materials can get into or out of the cell. Meanwhile, there are also peripheral proteins, which tend to be anchored to one side of the membrane, and these generally function as either enzymes or cell-cell junctions that help, those cell-cell junctions help bind and connect two adjacent cells to one another. There's also a number of carbohydrates that are associated with the plasma membrane, and these surface sugars are collectively referred to as the glycocalyx, which means sugar coat. So literally the plasma membrane of the external surface of our cell has this kind of forest of carbohydrates on its surface, and this forest is referred to as the glycocalyx. So let's start by looking at the lipids that make up the plasma membrane. There are generally two types of lipids that compose our plasma membrane. We have the phospholipids and we have cholesterol as well. Now, phospholipids form the basic structure of the membrane, while cholesterol serves to stiffen the membrane. So let's take a look for a second at phospholipids and what they are. So phospholipids, just by number, make up a majority of the lipids that are found in the plasma membrane. Now, their basic structure consists of this phosphate-containing head group and these two fatty acid tails that are joined to this phosphate-containing head group by a glycerol backbone. So we have a polar We have a phosphate-containing head group. I'll get to that term polar in just a second here, but we have a phosphate-containing head group, a glycerol backbone, and two fatty acid chains or tails that come off of the end of it. Now, this head region is referred to as polar, which means that there is a separation of positive and negative charges at the molecular level within the head group. So this makes the head group hydrophilic, which means water-loving. And so water tends to be, so I should say that this polar head group tends to face the watery environment either outside or inside of the cell. And so this is the water-loving or hydrophilic region of the phospholipid. Now the fatty acid tails, on the other hand, are not polar. And because they are not polar, that makes them hydrophobic, which means water-fearing. And so generally these fatty acid tails try to get as far away from the fluid environments as possible. So you'll tend to see the hydrophobic fatty acid chains within the very middle or core of the membrane. And so that's how they tend to organize themselves. So the reason why I bring up the terms polar and nonpolar is the fact that water is a polar substance. And so in chemistry, there is an old saying, like attracts like. So polar substances will be attracted to other polar substances. So since water is a polar substance, because again, this term polar in chemistry refers to the fact that there's a separation of positive and negative charges at the molecular level. So water is a polar substance. That phosphate-containing head group of our phospholipid is also polar, which means that water is attracted to the polar head groups of phospholipids and vice versa. And so the phosphate-containing head groups tend to face the watery environments, either inside or outside of the cell. And we can see that in this diagram here. So as I mentioned before, the plasma membrane is not just a single layer of phospholipids, it's a double layer or a bilayer. Because water is attracted to the hydrophilic polar head group, the hydrophilic heads of the phospholipids tend to face the watery environments, that is the extracellular fluid or the intracellular fluid, on either side of the membrane. And so you get the hydrophobic heads facing those fluid environments, and the middle or the core of the membrane is where the hydrophobic tails essentially kind of like to hide from the water. So those hydrophobic lipid tails, being water-fearing, so hydrophobic again just means water-fearing, tend to position themselves as far away from the water as they can get. So they occupy the middle of the membrane and face one another. So because of this, water is essentially excluded from the middle or the core of the membrane, and water does not easily cross from one side of the membrane to the other across this bilayer. And so the hydrophobic tails are really what allow the cell to separate the internal environment from the external environment. So the plasma, it's also kind of important to note here that the plasma membrane is not a solid structure, but it is a fluid structure. So the phospholipids that make up the plasma membrane do not behave as though they are solid, they behave as a fluid. And so because of the way that it behaves as a fluid, we describe the behavior of the plasma membrane by a model called the fluid mosaic model. And so what this model essentially highlights is the fact that the lipids and the proteins essentially are floating within the membrane. And so this means that the membrane is a dynamic structure. And the result of that is that it is a constantly changing structure or a constantly changing pattern. Pretty similar to the way that these plastic spheres are floating on this pond, right? So an individual sphere can move and change positions with respect to the other spheres that are floating on this pond. And so in much the same way, the phospholipids are doing the same thing in the plasma membrane. They're not stuck there in one place in the plasma membrane, but rather they can change positions with one another. And so the membrane is a dynamic kind of changing structure. So the phospholipids tend to be more fluid and move around the plasma membrane. Cholesterol is another lipid that we find in the plasma membrane, and it essentially kind of helps to stiffen the plasma membrane, so it kind of works against that fluid mosaic model that we talked about. So cholesterol is not as common as the phospholipids in the plasma membrane. Cholesterol generally makes up about 20% of the lipids that are found within the membrane. And cholesterol tends to kind of wedge itself between the phospholipids, kind of in close proximity to the hydrophobic lipid tails or fatty acid tails, because cholesterol, also being a lipid, is water-fearing, and so it wants to be as far away from the watery environment as possible. So it's going to associate really closely with the fatty acid tails that you see here as these little squiggly lines in this diagram here. Cholesterol actually functions to stiffen the plasma membrane. So what it does is it actually kind of links or binds adjacent phospholipids together, and so it helps to make the membrane a little bit less fluid in that regard. So we've just looked at the two different types of lipids that compose the plasma membrane. Let's turn our attention now to the carbohydrates that are associated with the plasma membrane. So as I showed you in the initial diagram, we saw that the extracellular surface of the cell has a thick coat of carbohydrate chains associated with it. Now, these carbohydrates can be associated with proteins, in which case they are referred to as glycoproteins. So this term glyco simply refers to, or simply means sugar. So these are carbohydrates associated with proteins, so we have glycoproteins. Or these carbohydrates can be associated with lipids, in which case they're referred to as glycolipids. And so collectively, as I mentioned earlier, the carbohydrate chains are referred to as the glycocalyx, and this term literally translates to sugar coat on our external surface. So note that the glycocalyx is only found on the extracellular surface. It's actually not found on the intracellular side of the plasma membrane. So we only have these carbohydrate chains on the extracellular surface. So the glycocalyx is a really important part of the plasma membrane. So important, in fact, that it's not just our cells that have a glycocalyx. Pretty much every cell that we know of, animal, plant, bacteria, has a glycocalyx on its surface. And you can kind of see it in this diagram here. It's highlighted with this little orange arrow here. You can see this fuzzy exterior on our bacterium here, and that is our glycocalyx. In this diagram to the right, the glycocalyx is this really dark, kind of fuzzy looking structure on the surface of the plasma membrane there. So let's look at the functions of the glycocalyx. So generally the glycocalyx functions in cell-cell recognition. So it's really important especially during development, that permits tissues to organize themselves, and so a developing cell has to know which tissue it belongs to and organize itself into a collective group. So a single cell needs to be able to associate with other cells that are similar to it and function in order to function collectively as a tissue. The glycocalyx also allows the immune system to recognize cells that belong to us, so this is referred to as self-recognition, and cells that do not belong to us. So things like bacteria, as I mentioned earlier, have a glycocalyx, and so our body is able to recognize something that should not be there, and if the immune system recognizes a cell that should not be there, it will destroy it. And so you can kind of think of the glycocalyx as kind of like a barcode for the cell. So it's an individual identifier or a marker that allows our immune system to tell whether that cell belongs to us or whether that cell does not belong to us. So it turns out that the glycocalyx plays an important role in the spread of cancer as well. And so a very kind of general definition of cancer at its core is that cancer is a group of cells, is a cell, or I should say a cell or a group of cells within our body that is undergoing uncontrolled cell mitosis, which is also known as cell reproduction or cell division, is occurring. And so anytime that you have a cell in which uncontrolled cell division or unchecked cell mitosis is occurring, that's typically referred to as a cancerous cell. Now, that's a very general definition of cancer, but at its core, that's really what cancer is. So, as you may be familiar with this term, metastasis. Metastasis is a term that we use when a cancer cell or a group of cancer cells break free from their point of origin and move to different places throughout the body. And these metastasized cells usually travel through the blood or the lymphatic vessels, as these are essentially the fluid conduits for the body. So it is thought that metastasis is possible because the glycocalyx of cancer cells changes and becomes much thicker, which makes it harder for the cancer cells to actually stick to other cells. And that then allows the cancer cells to break free from their point of origin, travel through the blood or lymphatic system to other places in the body where they can essentially set up shop. Because that's the other thing that cancer cells will do. Another thing that they will do is they will tend to seek out places where they can receive a lot of nourishment. And so cell mitosis takes a lot of nutrients, and so they generally, cancer cells try to generally flock to places in the body where they're going to get a lot of blood flow, and they're going to be able to promote the growth of a lot of blood vessels into that group of cancer cells. So one of the things that we also see the glycocalyx doing in cancer cells is the glycocalyx in cancer cells generally is continuously changing, and what that's thought to do is actually help the cancer cell evade the immune system, because as I mentioned earlier, the glycocalyx kind of works as a barcode, and the immune system gets kind of used to looking at certain glycocalyces or barcodes of cells, and so by constantly changing its glycocalyx, cancer cells can often stay one step ahead of the immune system. So this article that you see linked here, an image of this article here, how a thick sugar coating on cells may drive aggressive cancers, is actually a supplemental reading link that I placed in Moodle. So I would encourage you to take a few moments and read that article.